thing to be aware of. So an operon is a group of genes in prokaryotic bacteria that go and code for a series of proteins that carry out a particular task. Right? The Lac operon in particular goes and codes for 3 different enzymes. So Lac Z goes for beta-galactosidase, Lac Y for lactose permease, and Lac A, we don't need to know about Lac A. Right, so the genes, so the proteins, are only produced when lactose is present, because there'd be no point in producing it if there's no lactose present. So like we said, Lac Z codes for beta-galactosidase that breaks down lactose into glucose and galactose. The bacteria can go and then use the glucose in respiration. So normally, if there's no lactose present, then the repressor protein is bound to the operator and the promoter region. When lactose is present, it binds to the repressor protein, to an allosteric site, causing a conformational change. Right? This conformational change causes it to change shape, so it can no longer bind to the operator and the promoter, so it breaks across. Right? This enables the RNA polymerase to go and move all the way down and start transcribing the 3 genes. If we're looking though at increasing the amount of these 3 proteins that are being transcribed, we need to go and have a promoter. Right, normally when glucose is present, this cap protein, which is the promoter protein, is inactive. So to activate it, we require a molecule called cyclic AMP. So we produce cyclic AMP using the enzyme adenosine cyclase. When glucose is at high concentrations, that inhibits adenosine cyclase, right? So it causes it to be inactive. When glucose isn't in high concentration, that causes the adenosine cyclase to not be inhibited, so therefore it's activated. When adenosine cyclase is activated, it breaks down ATP into cyclic AMP and 2 inorganic phosphates. That cyclic AMP goes and binds onto an allosteric site on the cap protein, which is the promoter, which causes it to go from being inactive to active. The cap protein will go and bind onto the operon, somewhere between the operator and the promoter, and it will increase the binding strength of the RNA polymerase to the operon, so therefore more of the protein will be transcribed.